Okay, so today we're going to pick up from where we left off yesterday. We're talking about linked genes, genes that are on, different genes that are on the same chromosome. And so during meiosis, we see that when the chromosomes, the homologous chromosomes separate, whatever genes happen to be on that particular chromosome all travel together. Okay. However, there are some differences, or there's some exceptions to that rule. If we remember when we talked about meiosis, that when homologous chromosomes are together during metaphase, some of their chromatids get all mixed up, and so we have something that is referred to as crossing over that takes place. And so during crossing over, what we see is we have homologous chromosomes side by side, and so part of each of the chromatids from homologous chromosomes will jump chromosomes or will switch places. And so this just increases the variation within different species so that we're not all identical, and kind of mix and matches the genes. Now, where the crossing over takes place is going to be different from chromosome to chromosome. Sometimes it's way up here by the center or the centromere, and sometimes it's just the tips that get exchanged. But notice here that when we have crossing over, that it is only one portion of one chromatid that actually gets exchanged. Okay, so this crossover of the chromosomes is going to throw a kink in kind of how we, or what we expect for our genetic crosses when we do our Punnett squares and figure out the probability of these genes and how they travel and what we expect the offspring to look at, because we're not going to see these nice 9:3 to 3:1 ratios, or we're not going to see the 1:2 to 1 ratios that we expect to see. So we can use this information and this crossover frequencies to try to figure out where these genes are located on various chromosomes. And so we do that by creating a gene map. And this is going to use something that's called recombination frequencies. So how often do these genes recombine on different chromosomes in order to give us the traits that we see? So depending on how often these genes are going to recombine is going to tell us how close to or how far away from each other the genes are on the chromosomes. The closer that two genes are, if we take a look down here at this example, okay, the closer that two genes are together, the less likely they are going to separate during meiosis or separate during crossing over, because they are so close together. I keep losing my mouse, sorry. If I take a look over here on our second example, if the genes are farther apart, then it's a little bit more likely that they will separate during crossing over, because I have a lot of space in between them that will allow for the opportunity to separate the genes from one another. So we don't have the big A and the big B on one chromosome and the little a and the little b on another. The big A might switch places with the little a, and so we get that recombination there. Okay, so link maps are going to show us where these genes are in relationship to one another on a particular chromosome. All right, so how do we do that? So we do know that each gene is found at a fixed location on the chromosome. And so we can take these maps and we can use them to identify where they are, which is going to then further help us study them better. Okay, we can use these locations to clone the genes so that we can better understand what they do and why they cause genetic diseases when they mutate. Okay, it also is going to allow us to go in and start inserting the good genes with the bad genes. Once we can identify where they are, we can sequence them and we can start cutting them out and replacing them. All right, when we talk about linking the genes, we call this a linkage group, and these are all of the genes that are on a particular chromosome. And so when we go and we map these out, we will call this a map unit or a linkage map unit. All right, and it'll look something like this. Okay, where we have the B gene is 6 map units or 6 linkage map units away from this PR gene. And the PR gene is 12 map units away from the W gene. All right, so we're going to do a little example here. So let me switch screens. get the right buttons. Okay. All right, so if I am given the information that we see that there's an A gene, a B gene, and a C gene, okay? And so we've gone through and we've calculated how often do these genes cross over from one another, okay? And we'll talk about how to do that in just a minute. So we go through, and what we're going to see here is that the A gene and the B gene cross over or do not show the patterns that we expect 8% of the time. And so what we do know is that 1% is equal, so 1% when we're talking about the frequency of crossovers, is equal to 1 map unit. Okay, to 1 map unit. And we'll see it abbreviated as an MU and sometimes as an LMU for map unit or linkage map unit. All right, so when we see this, what we're going to do is we're going to try and figure out where these genes are located in respect to one another. So we know that I always like to take a look at the biggest crossover frequency, and that is going to be the B and C gene. So I know that the B and C gene are the farthest genes apart. So I go ahead and I put this information down. I'm going to put the B gene here, and I'm going to put the C gene here. I could easily put C here and B here. Doesn't really matter. And so we know that this is 10 map units away from each other. All right, and then I'm going to start to look at this. So I come back up here and I see that the A gene and the B gene are 8 map units, or 8. They cross over 8% of the time. So they're 8 units away. So down here is the B, and so when I go to find the A, it's going to be approximately 8 map units away. Okay. Now I'm going to double check that, and I'm going to look here from the A to the C should be 2 map units. And so I get here. If I add these two up, 8 and 2, I should get 10, which I do. All right, so given the frequencies here, this is how I would figure out where these genes are located in respect to one another on a chromosome. Okay, so now we're going to take it a little bit further. So how do I get those frequencies? All right, so we're going to do a little example here, and what we are going to do is we're going to work with 3 different genes. And on these genes, what we're going to see is that we have a green plant body is going to be denoted with the letter C. Okay? And then the wild type, or the dominant allele, a lot of times what you'll see geneticists do is that they're going, hold on, let's move me. Okay, so what the geneticists will do is instead of dealing with big letters and little letters, they're going to switch it out and use a plus. So they'll use the little letter to represent the recessive allele and the plus to represent a dominant allele, which is also called the wild type. Okay, so my recessive allele is going to be for a green plant body, and the wild type, or the dominant allele, is going to be a plus, and it's going to represent purple. Okay, the BZ gene is going to be the bronze seed, and that's going to be the recessive allele, and then plus is going to represent the purple seeds. So WX is for the waxy endosperm, and endosperm is the food source for the plant, for the embryo as it's growing. And then the plus is going to represent starchy endosperm, which is the dominant wild type. Okay, so we're going to take a look at these 3 alleles here, and I'll give you a little background on what happened. So the first thing that we did is that we took a plant that looked like this. All right. And so I have all 3 of these alleles are going to be present, and we're really just representing the genotypes here. And because I only have single letters, this is going to represent that I'm big C, big B, big bronze, and big wax. So it's homozygous dominant or true breeding. So this here is my parent generation, my P generation. I'm going to take this true breeding plant, and I'm going to cross it with a plant that is wild type for all 3 dominant alleles. Okay? So I have a homozygous recessive plant, and I have a homozygous dominant plant for all 3. And so they're going to come down, and they're going to give me my F1 generation. All right, so another way to write this is that we would write CBZ, WX, and then a plus, plus, plus. And because I have this slash mark here, this is representing that I have a heterozygous combination. It's a C with a plus, a B with a plus, a waxy with a plus. All right. So when I have a heterozygous combination here, what researchers did is they took this and they crossed it with the same type. Okay, so they crossed it with another heterozygous. And so the expected ratio that we expect to see is a 9:3:3:1 ratio. All right, so when researchers went through, or about that, so when researchers went through and they took a look at this ratio, what they found out is that this is not what they got at all. All right, and so how would we confirm that? Well, we would perform a chi-square test to confirm. All right, so what we found after doing the chi-square test is that we are not falling into the ratio that we expect to see. And so what we would do then is we're going to try and figure out, we would assume that these genes are not being inherited independently from one another, so therefore they are on the same chromosome. So we want to try and figure out where exactly these chromosomes are located. And so these genes are located on the chromosome. So the first thing then that we're going to do is we are going to take one of these F1s, okay, and we are going to perform what's called a back cross. Okay, sometimes it's also referred to as a test cross. Cross. And so when we do a test cross, what we are doing is we are going to mate it with a homozygous recessive individual. All right, so my cross is now going to look like this. CBZ, BZ, WX is going to be crossed with my heterozygous wild type, my heterozygous plant. All right, and then I'm going to calculate out, or I'm going to go through and I'm going to count the different phenotypes that I see. Okay, so when I do this cross, these over here are the results that we actually saw. Okay, so now we're going to go through, and now that I have that information, we're going to go through and we're going to try and figure out, now how do I calculate the frequency of crossovers? So I'm going to switch papers here. All right, so here's my data. And so the first thing that I'm going to do when I perform this linkage map or try to figure out the frequencies is that I have to know what the P generation or the parent generation is. And in our case, we were homozygous for all three traits here, and we were homozygous for all three traits here. Okay, now another cue in this information is that your parent group, your P generation, is usually always, unless they state, it is always the group that occurs most often. So this is what we would call normal. Okay, so I need to know what my normal groups are, or my parent generation. So here we are with the homozygous. So I'm going to go through and I'm going to say, all right, well, I'm going to start comparing two genes to each other. So I'm going to look at the C and the BZ gene. Okay, and so I need to know what normal is. And so in this case, my normal is a C and a BZ for my parent, or they were plus plus. So when I go through, I'm going to look for individuals that do not fit this pattern of a CBZ or a plus plus. So I have CBZ, and I've got a plus plus here. I have a C plus. This does not match what I have. Okay, a plus BZ does not match what I have. A C plus does not match, and neither does a plus BZ does not match. Okay, so now what I'm going to do is I'm going to take a look at these individuals that do not match what my original parents were, and I am going to go through and I am going to add them up. Okay, then I'm going to pause this while I do this because I'm running out of time. Okay, so here we go. I finished up the information. So what I have done is I have color-coded the blue with the blue. So again, I'm looking for the patterns that do not match what my parent groups are. I'm going to add those numbers up. Then to find the frequency, I am going to take my totals and I'm going to divide it by the total number of total offspring. Okay. Once I get that, 45 divided by 900 is going to be a 5% crossover frequency. So once I have all of my crossover frequencies identified, I can take that information and I can go ahead and plug it down, or plug it into a map group down at the bottom. Now, one of the things that you will notice